Good morning. My name is Harshit Khatri and I am a student of MSc DBT Biotechnology second sem. Today in this e-video I will be talking about the major histocompatibility complex, and mainly I will be focusing on exogenous antigen, that is endocytic pathway. So first of all, what is major histocompatibility complex? Major histocompatibility complex, it is a group of tightly linked cluster of genes, and MHC is located on chromosome 6 to humans and chromosome 17 in mice, right? The MSC referred to as HLA complex of for human and H2 complex for the mice. And MSC genes are synthesized into or organized into three complex, that is class 1 MSC, class 2 MSC and the class 3 MSC genes. Class 1 and class 2 MSC genes encode MSC protein that present inside that present antigen to the T cells, right? So in human, class 1 MHC genes HLA, HLA-B and HLA-C, they are mainly DP, DQ and DR. And class 1 MHC protein express glycoprotein that is 43 kDa size and alpha 1 and alpha 2 and alpha 3. Class 2 protein that is 35 kDa and alpha and beta polypeptide cells which are presented in this, that is CD8 T cells, right? So if we talk about the procedure, how the antigens taken processed inside the RER and how the process is going to take place. So it is an exogenous antigen, that is endocytic pathway. First of all, what happen here is कि antigen presenting cells require exogenous protein antigen by phagocytosis or pinocytosis. Then proteolysis is required to convert the antigen into peptides for the binding of the MHC2 molecule. Then proteolysis occur in the acidic medium, that is endosomal and lysosomal compartments. A wide range of peptides released or produced and for the binding of the MHC molecule, right? So when class 2 MHC molecules synthesized within the RER, they associate with the protein called the IV chain. It serves as a chaperone, right? So the IV chain is cleaved by proteases is the endosome in the late endosome, and however, a short fragment of the IV chain is termed as clip, which is remain bound to the MHC2 molecule. The clip is and here in before after going to the Golgi complex, the clip is resistant to the proteolytic attack. So at last, the fragment is released and freeing the MHC protein to bind the peptide mediated by a non-classical class 2 MHC protein that is called DM. So class HLA-DM mediates the exchange of clip for antigen peptides. So if we talk about the summary of how this process takes place. So first of all, the class 2 MHC alpha and beta bind to the invariant chain and blocking binding of endogenous antigen. And MHC complex is routed through the Golgi, right? Golgi to the endocytic pathway compartments. The invariant chain is degraded, leaving clip fragment. And the fourth, exogenous antigen is taken up and degraded, routed to the endocytic pathway compartments. HLA-DM mediates exchange of clip for antigen peptide, and at last, the class 2 MHC peptide is transported to the plasma membrane. Thank you.